my name is Jayden, and today I'll be talking about macromolecules. So what are macromolecules? Macromolecules are large molecules that are essential in biological processes. There are a total of 4 types of macromolecules, including carbohydrates, lipids, protein, and nucleic acids. Before introducing you guys about macromolecules, I would like to talk about what polymers and monomers is, which is consist macromolecules. So polymers are long chain-like molecules consisting of many similar building blocks called monomers, and all macromolecules do have polymers and monomers except for lipids, which I will talk about. So how do you make polymers? So to make polymers, you need to go through something called the dehydration reaction, which is when 2 monomers bond together to dilute loss of water molecules. So as you can see in the picture below, glucose adds with glucose while losing an H2O, make maltose. On the other hand, you could also break down a polymer to make 2 monomers, which is done by a process called hydrolysis. And the first type of macromolecule I'll talk about is carbohydrates. So the function of carbohydrates is it gives energy and fuel for our cells. So for example, the reason why I'm here and talking is because I probably have carbohydrates in my body, and monomers of carbohydrates are called monosaccharides. The chemical formula of monosaccharides are CH2O times N. Examples include glucose, fructose, galactose, and the formula of them are C6H12 and O6. You could also differentiate glucose from galactose. The most important difference is that fructose is a pentatone, while galactose and glucose. So a more complex version of monosaccharides are disaccharides. They're a combination of 2 monosaccharides. Its chemical formula is C12H22O11. It's not C12H24O12 as many would expect, but it loses an H2O because of the dehydration reaction I talked about. So it makes this kind of formula. It is formed by a covalent bond called a glycosidic linkage. Examples of disaccharides include sucrose, lactose, and maltose. A more complex version is called polysaccharides, which is a polymer version of carbohydrates. In function, there are 2 major types of functions, which are storage and structural roles. So first of all, for storage, there's starch, which is related to plants, found in chloroplasts and plastids, and glycogen, related to animals, which is found in liver and muscles. For the structure role, there's cellulose, which is the component of plant cell walls, and chitin, which is found in the exoskeleton of arthropods. One of structure of these polysaccharides is that there's 2 ring forms called alpha, which is helical, and beta, which is straight. As you can see in the table above, while cellulose is a beta glucose, which is why it's straight. Amylose, amylopectin, and glycogen are all alpha glucose. Now I'll talk about the second type of macromolecules, which are lipids. One characteristic is that it's hydrophobic, which means that it doesn't like water. The reason why it's hydrophobic is because it is consisted of hydrocarbons, which form non-polar covalent bonds. As I briefly talked about, it do not form polymers because it doesn't have monomers in the first place. There are 3 important types of lipids, which are fats, phospholipids, steroids. First of all, fats. Its function is for energy storage, and it's constructed of glycerol and 3 fatty acids. Glycerol is a 3-carbon alcohol with a hydroxyl group, OH, attached to each carbon, and fatty acid, the long hydrocarbon chain. There are 3 types of fatty acids, which are saturated fatty acid, which do not have any double bonds, and examples are animal fats, unsaturated fatty acids, which do have double bonds, and oil. The last one is a trans fat, which is quite unique because the result of hydrogenation. This is when you add hydrogen molecules into an unsaturated fat to make saturated fat, and it kind of make a trans double bond. The bond between glycerol and the fatty acid is made by ester linkage, which is also a covalent bond form fats, and fats are also called triglycerides. The second type, phospholipids, it consists our membranes in our cells, and consists of two fatty acids and a phosphate group, which is attached to the glycerol. It has a hydrophilic head and a hydrophobic tail, which is why it's called an amphipathic molecule. And this kind of thing results in a bilayer arrangement in cell membranes because when it comes to water, while the head wants to face the water, the tail kind of runs away from it. The last type is steroids, which is made by a four-fused ring made of carbon skeleton. An example is cholesterol. As you can see, there are 3 hexagons and 1 pentagon. The third type of macromolecules are called proteins, which is also known as polypeptides, which is the polymer version of proteins, and is built from a set of 20 amino acids. As you could guess, these amino acids are the monomers of proteins, and it is consisted of a carboxyl, which is COOH, and an amino group, NH2, attached to the central carbon. Also, below the central carbon, there's a R group, which kind of makes lots of different amino acids. So, because there are 20 different types of R groups, you can infer that there are 20 amino acids. This is made by the peptide bond, which is the CN bond, as you can see in the picture below. And examples include enzymes, which are very important in our body because speeds up reactions. So there are 4 levels of protein structure, which is probably because the difference in structure, like 2D and 3D. So the first one, primary structure, it's a unique sequence of amino acids. So think of it just as a line with lots of amino acids. The second one, secondary structure, which is a regular coils and folds made in polypeptide backbone. And this is made by the hydrogen bond, and there are 2 types, alpha helix and the beta pleated sheet. Third type is tertiary structure, which is shaped by interactions among various side chains of R group. These include lots of bonds, including covalent bond, hydrogen bond, van der Waals, interactions, and more. The last one is a quaternary structure, which is made of multiple polypeptide chains, and it is the most complex version of proteins. It includes many of the proteins we know, such as collagen and hemoglobin. So one important thing about proteins is that alterations in pH concentration is catastrophic. It's called denaturation. The sickle cell disease is the result of denaturation, which is very bad for our body. Now I'll talk about the last form of macromolecules, which are nucleic acids. The polymer is a polynucleotide and is made by phosphodiester bonds. And one thing about nucleic acids is that think of it as a line and then think that while one endpoint is always a 3', the other endpoint needs to be a 5'. I'll further talk about that soon. So the monomer of nucleic acids are nucleotides, and it's consisted of a nitrogenous base, pentose sugar, and phosphate group. There are 2 different family groups of nitrogenous base, which are pyrimidines, including cytosine, thymine, and uracil, and purines, which are adenine and guanine. And as many know, the function of nucleic acids are the instructions for genetic information. So there are 2 types of nucleic acids, which are DNA, also known as deoxyribonucleic acids, which is responsible for its own replication and synthesis of mRNA, and RNA, which is also called ribonucleic acid. So first of all, the DNA. Its pentose sugar is in the form of a deoxyribose. It doesn't have an OH compared to the RNA, and then the DNA structure is a double helix, as you can see in the picture, right? It's kind of like folded constantly. And it is consisted of 2 backbones, which run in opposite 5' to 3', as I talked about. And because of this structure, it is called an antiparallel. And between the nitrogenous bases, pairs are made by the hydrogen bonds, which include the adenine and thymine, they bond together, and then cytosine and guanine, which bond together. And the second type of nucleic acids are RNA, different from DNA, it's one-stranded, and the sugar is ribose. And its nitrogenous bases include cytosine, guanine, adenine, and uracil. One important thing is that this RNA doesn't have thymine, but instead it has uracil. So there are a few different types of RNA, which is tRNA, rRNA, and mRNA. Their functions include like transporting ribosomes and possibly like making proteins. So why do we need to know these macromolecules? First of all, it's fundamental to our lives. And second of all, it helps us understanding of biological functions, contributes to advancing biotechnology, particularly in fields like medicine. So yeah, that's all of it. I talked about macromolecules and I wish you know about it. Thank you.